Thank you once again for taking part in today's virtual talk with 3 exciting presentations on the topic Innovative Technologies for Pharmaceutical Production with the special Chinese pharmaceutical industry. So we also look forward to providing you with further insights into many other topics at Pautec Technofarm in Nuremberg from the 23 to 25 September 2025, so this year. So make note of this date. And also for your information, with our reactivation of Technofarm, we want to offer you a platform for the growing importance of the pharmaceutical and life science industry. So many exhibitors from this sector have already registered for Pautec Technofarm in Nuremberg, and we also have chosen this virtual talk topic today to match to this industry. So in generally, our talks will continue to feature exciting topics and experts from a wide range of industries. So, and I would like to take this opportunity now to thank our speakers from today, Philippe Steinbusch from AZO, Dr. Helena Weingrill from Anton Paar, and also Dr. Andreas Risch from Fette Company in China. So without them, this would not have been possible today. So now I would like to hand over to Martin Bornhöft, Managing Director of APV. As an honorary sponsor of Pautec Technofarm, the APV has been an important partner for us for many years, and we are looking forward to our further cooperation and exciting projects together with APV. So, dear Martin, thank you, and the stage is yours.
Thank you very much, Clara. A warm welcome from my side too. I'm delighted that you're here today. For all those who do not know APV yet, the APV is the International Interdisciplinary Independent Professional Association for Pharmaceutical Technology and Industry Pharma. As Clara mentioned, we are a close partner and conceptual sponsor of Pautec Technofarm, and we are also in constant dialogue as a member of the advisory board. We work together to continuously develop the fair and adapt it to the current needs of the industry. This year at Pautec Technofarm 2025, we are expecting around 600 exhibitors and around 12,500 visitors. We want to offer an exchange between expert and a knowledge transfer from the pharmaceutical industry. Through formats such as virtual talks, which we have now, or on-site stage talks like our Technofarm Forum with exhibitor and invited talks, we can promote this exchange and present solutions. In this context, I would also like to mention the Pharma in Focus joint stand on-site at Pautec Technofarm this year. Use this joint stand as a company from the pharmaceutical process engineering sector to enable simple and uncomplicated participation in the trade fair. It's an all-inclusive package that you can make your first step to Pautec Technofarm. Please make a note of 23rd to 25th of September 2025 in your calendar. I look forward to meeting and welcoming you in person at Pautec Technofarm. If you have any questions, please feel free to contact the team or myself. And now we are coming back to the virtual talks, which are also an example of Pautec Technofarm's commitment to making expert knowledge accessible and promoting dialogue within the community. We expect exciting presentations and discussions. So I do not want to talk too long, and I'm looking forward to the first talk by Philippe Steinbusch from AZO and his talk about pharmaceutical powder systems for robot-assisted API production. Philippe, the stage is yours. Enjoy the talk.
Thank you, Martin. I will share my screen. So, thank you also from my side for joining the presentation virtual talk. First, I present myself. So, Philippe Steinbusch, I'm a sales engineer for pharmaceutical and containment applications. I would like to present you a little bit an application for the API production within the pharmaceutical sector and how we can robotize or automatize these systems. But first, I would like to present you a little bit who is ASO. So ASO is a global market player for powder handling systems and semi-liquid and semi-solid systems. We are a family-owned business created in 1949 by Adolf Zimmermann, and we are still a family-owned business located in Germany. We have more than 1,000 dedicated people to supply solutions in powder handling and semi-solid and semi-liquid handling. German company, we have still our own production sites, still located in Germany. We have actually 2 production sites. One is taking care of the raw material handling, solid handling systems, and the other one is taking care of the semi-liquid, semi-solid systems like homogenizing, dispensing, and mixing liquid with solids. We are located throughout the world in different locations. We have an office in North America, in China, in Asia, and of course several offices in Europe. If we take a closer look to Europe, we have a location in the UK, in France, and in Belgium for the Benelux market, and of course our 2 production sites together with the headquarters. What does ASO do? We make machines, process lines, or complete production lines for solid handling and semi-liquid handling. So this means, for example, all the raw materials coming into your plant, is it in bulk, is it in big bags, is it in drums, or is it in bags? We take care of them, we store them, maybe intermediate storage, or we dose or we weigh them directly to the core, the heart of your business, which can be a mixer, an extruder, a dissolving system, a dispensing system. It really doesn't matter. We take care of all these flows, we transport them, we dose them, and we weigh them. And if needed, we can also take care of the flows afterwards once the process is done for packaging lines, for example. Of course, we do not only make machines or equipment, we also make a complete production and process lines, meaning that we can take care of the operating and controlling system, as well as the whole electrical part and software programming of the system. So this was a little bit the introduction of ASO, but all these process flows, if we take them to a pharmaceutical application, for example, if you want to make OSD, you have different smaller components, and how can we, for example, use robots to take care of these process lines? Well, I want to share you today an application we did for one of our customers, and the application was actually this first requirement, which was this vessel, special vessel, which needed to be filled. We needed to bring it to a mixing system, and then we needed, of course, to discharge it. So what were the requirements? First, customer wanted an automated production. We needed a lot of availability, no stops, not related to operators, and we needed to have really a continuity and to have a continued production. Second, we needed high precision. As you know, we are talking about pharmaceutical components, so we need to be careful that the amounts are highly weighed in a high precision. Of course, ergonomics is important. Operators cannot lift too much, so we need to take care of that as well. Second, the next step was containment and ethics requirements. As you know, that most of pharmaceutical products are not easy in terms of containment, and also they are very often explosive. We need batch integrity, so we need cleaning of the equipment. We need traceability. We need to know what's going inside, what's coming out, and is it mixed correctly. And finally, the software needs to be compliant according to GMB regulations. So what was the idea? We had this vessel, and how are we going to handle this vessel? This vessel, you need to know that it's very heavy. It was around 100 kg, so it's not so easy to manipulate. So the idea came to use a robot system to do all the stuff and to have an automated production. So before I show you a little bit of details of the different equipments, I want to show you this video. And this video was recorded during validation, and I speeded it up to show you a little bit the movement. So it's not the real movement, it's just to show you which movement the robot does during the whole production cycle. So here you have the robot in the middle. He scans, you see that a green light, he can scan a certain vessel with a barcode. He takes a vessel and places a vessel on a mixing stand. During that time, of course, he's free and he can move around. He can take another vessel. In this case, he scans again the vessel. He wants to be sure that he has every time the right vessel, and he can place it on another position where, for example, here the product would discharge into another position. I will explain later how these different positions work and what they do. Then, of course, the robot takes another mixing vessel from another position and can move it freely around. So as you can see, you can have different mixing vessels in that area, and the robot takes care of all the movements. Finally, a last movement is that the robot takes the empty vessel again and can place it again on the in the filling position. So if we go a little bit in detail, we have the different raw materials. We need to add them to the mixing vessel, and the raw materials, in this case for this customer, there were 3 raw materials that needed to be mixed together, marked by 1, 2, 3. And we have 2 filling positions, so we have a position A and a position B, and the robot can place a certain vessel on a position A or B. These raw materials needed to be dosed, so what we did, we used feeders, dosing machines, and we can, for example, we have 3 dosing machines. They can fill together. 2 feeders can dose in point A, and 1 feeder can dose in point B. And we have a very high accuracy. So these feeders can dose at a 5-gram accuracy or even better. And the feeders dose into a hopper, and this hopper has also, this hopper has weighing cells, and has also an automated calibration system. So before we dose, we calibrate the whole system again to be sure that we have really the correct amount, and then we can dose with a high accuracy. Once the product is dosed, we transfer the whole product into the mixing vessel, which is located underneath. These dosing systems have very small volumes. As you can imagine, if you want to produce during a certain time, for example, a few hours, you don't have enough product in these feeders. We didn't want to make the volumes bigger to have a very correct amount, a very high accuracy. So we will refill these 3 feeders with the chute tubes, which are located on where you see the 1, 2, 3. The refill system is located on the level above. So if we look into detail how it looks like above on the first floor, we have these different filling systems. We need a certain buffer because we want to have as less possible operators around and taking care of the system, because we need a very high availability, and in taking care, because we also need to have a cleaning of the whole system. So once the production is completely finished, we can dismantle every product contact piece and bring it to a washing area. But that means also that if you have a buffer, for example, you cannot easily remove a buffer. So in this case, we used an IBC as buffer, which is placed on a certain fixed position, and we can completely fill this IBC, which is then the buffer for a certain number of hours. And once it's empty, or when we are finished with the production, we just remove the IBC and we can clean it in a washing street or a washing area. Here you see, for example, that we need to refill them. And usually when you talk about pharmaceutical products, they come in as raw materials, and these raw materials are usually in drums or bags. So we have position 1 and 2. They are filled from drums. And how do we fill them? Because it's not so easy to manipulate drums. We have designed a lifting column which has a clamping system. So basically you only place a drum in a clamping system. the lifting column clamps the drum, lifts it, rotates it, and swivels it to the position. And by means of a split valve technology, we can connect the drum to the IBC. And so in this case, you see on the video in the middle, he's finished swiveling, then now he rotates. And then finally he will dock to the IBC. And we can empty completely the drum. So this helps for the ergonomics for the operator. The only thing he has to do is to open a drum and place it in the clamping system. The third product was filled by means of a conveying system. So we just take a suction lens and we can empty the bag into an IBC. So this is how the raw materials were managed. So operators have less effort to refill the system, and the system refills itself and doses it itself in an automatic way. We have dosed the product, it goes to the mixing vessel, it goes to the mixer, we're finished, and then we need to empty this mixing vessel into the next step. This mixing vessel was emptied in an IBC, and this IBC can contain 4 to 6 times a mixing vessel. So once this IBC is filled, it gives a signal to the operator, and the operator needs to replace the filled IBC by an empty one. So in order to avoid the robot to stop, because we cannot enter in the room, so the room is completely closed so that no operator can enter, we designed a system to access to the IBC, but without the robot stopping. So the robot can continue cycles. And from that door, as you see there, the operator has access to change the IBC. So I have 2 doors. The IBC is placed on a weighing scale so that we can monitor how much is inside. And once the system gives a signal to the operator to change it, which is then happening every 4 or 5 hours, we replace the IBC. Once this is done, of course, the mixing vessel, in this case, it was a requirement to clean it between each mixing step. Before refilling it again, we needed to clean it, to sip it. And there also, we cannot have an intervention of an operator. So the robot places the mixing vessel into a washing station. And this washing station contains also a split valve technology so that it can dock automatically, it can clean it, it can sip it, and then dry it again so that the mixing vessel is ready to use again on the first filling stations, which is A and then B. So this is the complete cycle of the robot, and he does this. He can just do this endlessly the whole day. What you need to take care of is, of course, that sometimes it can happen that you have a non-conformity of a mixing step or a washing step, and so you need a position where you can remove a vessel. But also when you start your cycle, your production cycle, you need to introduce a lot of vessels, so you have a fixed position which is needed to remove or place new vessels. So this concludes a little bit the different steps. And if we look into the requirements which was requested by the customer, we have the automated production cycle, which is okay. So all the steps what you see here could be managed. So automated production cycle, because the robot does all the job. We have a high precision weighing. We also have an automated calibration system. Ergonomics, the operators only need to place drums on the clamping system or empty a bag with a suction lens. So it's really easy for the operators. Containment and ethics requirements, because we have a completely closed system, we have a certain containment level, and ethics also, it makes it easier because we have ethics requirements on the inside of the system, but on the outside it's limited because the system is completely closed. Batch integrity, due to the fact that you can sip the intermediate mixing vessel after each cycle, we can guarantee a certain integrity, and also we can clean the whole system so everything is dismantleable and everything can be cleaned separately. So we cover that step. Batch traceability, as you could see, the robot has its own scanner on the arm. So at each step, he can scan the vessel and he can check, he can double check if he has the right one and if the the movement he does is correct in function of what needs to be done. The whole system was covered by an ASO automated software, and we are GMP compliant, so the software was 24 CFR Part 11, so we could cover the whole GMP regulations with that. I hope I could give you an insight of what is possible in API productions. And of course, this is an application for one customer. As you can imagine, every customer has its own process steps, has maybe more ingredients or less ingredients, has another mixing system, has another part where he wants to fill or empty the vessels. So everything is possible, but this gives just an insight of what is feasible in the API world. I want to thank you for the presentation, and if you have some questions, I will try to ask them, to answer them as much as possible.
Thank you very much, Philippe. So you are now free to ask your questions over the F&A or Q&A button at the bottom of your screen. So I will give you a few moments that you can type in your questions, and then I will forward it to Philippe. So we wait some moments. So first question, has the process a standard configuration, or is it possible to adjust it in function of the customer needs?
So it's adjustable, so there's no fixed configuration. Of course, you need to take care that the robot has a limited range. So we have a limited space around the robot, and you need to work into a circle. So as long as we don't need more than what's feasible in a circle, we can do whatever we want.
Thank you very much. So next question, is a PAT system integrated? Can you take samples for IPC?
In this case, it was not integrated, but it would be feasible. But then we need to discuss where you need to take the sample and how much you need to take the sample. But there are possibilities to integrate a sampling system, yes.
Thank you for answering. Perfect. Well, yes. Which clean room class is required for this process?
So we are not in a sterile environment, so we are talking about OSDs. So because it's a closed system, you could eventually think about non-classified, controlled but non-classified room. But I would suggest that classroom D is at least necessary. Going higher, I'm not sure it makes sense because we, as I said, it's not a sterile environment. But it's up to you. But the higher we go, the more we need to be careful with the equipment we place into the rooms.
Okay, thank you very much. So now we do not have any questions left. So if you have any questions, please feel free to ask them now. If you have questions afterwards, please feel free to contact also pautec-technopharm@nürnbergmesse.de. I can forward it also to our speakers and maybe match your contact so then you can talk to each other personal. So if no questions are left, I would say a big thank you very much to Philippe for your interesting presentation, and go then forward to our next talk. So now we continue with Dr. Helena Weingrill from Anton Paar and her talk about powder rheology as the versatile tool for enhancing any pharmaceutical process. So thank you, Helena, for joining us today, and have fun.
Thank you very much for the introduction. Give me 1 second, I'm sharing my screen. Okay, great. It should be visible. So thank you again for handing over. A very warm welcome also from my side. As mentioned, we will basically talk how powder rheology can be used for improving any pharmaceutical process. And of course, here I am referring to pharmaceutical processes where granular material is involved. So let's see what this is all about. Sorry, fast forwarded. So before I jump into the topic of powder rheology, what I would like to discuss before that is actually not powder rheology, the entire term, but rheology per se, because I expect a couple of people in the audience who are not particularly familiar with this, let's say, rather exotic field of science. So rheology, hold on, give me 1 second. Rheology is the flow of matter of any material, actually. This is what we are studying with rheology. I am saying here any material because really anything can flow. So obviously liquid flows, right? Solids flow and so on, but also powders flow. This is why there's this need for powder rheology. This was already stated more or less by this Greek philosopher Heraclitus, which you can see on the left-hand side. He said pantere, and exactly this is where this word actually comes from. If you're looking at this ray, this is exactly where rheology, the word stem comes from. Well, as we see, he's sitting right next to two different liquids, which are rather delicious. You can decide which one is the more delicious one. And what we know of these liquids is that both flow, right? If you tilt over the glass, the beer for sure will flow out, right? As Heraclitus does it here. However, if you do the same thing with honey, well, it will also flow, but it will do this at a different pace. And this is exactly where rheology comes into play, because here we actually need to quantify these differences in the flow behavior. And this is actually where Heraclitus, with his famous saying, pantere will not help you anymore. And he would tell you to get a rheometer for quantifying these rheological characteristics. And this already leads me to my next slide. I would like to quickly explain what a rheometer does. Well, basically, a rheometer forces a material to move. It can do this either in a linear way, in a rotational way, and so on. And it measures at the same time the effort which is necessary to make the material move. And by measuring that, what we get out is viscosity, right? Viscosity is this term which determines my flow behavior, and we get many more results. However, viscosity is the most important part to get out. Let's take a look at the hardware, how this is done. Well, we see our rheometer here. The heart of the rheometer is always its head, because in here we have an engine, right? And this engine on the one hand forces my material to move, and on the other hand, it detects the signal, which is then used for calculating the viscosity and other magnitudes. To the engine attached, we have a measuring system, and this is also of importance because this measuring system will have to be adapted to the material in interest. I've mentioned that we can measure how solids flow, how liquids flow, and also how powders flow. And you can easily imagine that we would need different measuring systems now to, well, make a powder flow as opposed to make a liquid flow. Well, besides those two parts, which are definitely the main part always of rheometer, so the engine and the measuring system, what we can adjust as well or add as well is always some surrounding temperature with a temperature device, a humidity generator. And this is one of the most important takeaway messages of today. You always have to set the surroundings during the measurement to exactly what you see during the process. So in general, powder rheology is about imitating powder handling processes. But if you're not setting the temperature right, humidity and so on, the applied pressure, you will never get representative results. So please keep this in mind. Flow behavior heavily depends on the temperature and the humidity the material is seeing. And of course, we can add our powder cells. So let's actually see why it is necessary to look at the flow behavior of powders themselves. The thing is, there are so many impacts that let powder flow differently. We can categorize them into internal impacts and external ones. Let's start with the internal ones. Those are basically the impacts where we as well, let's say powder rheologists, can do nothing about it. Those are the given, the physics from the particles that make up the powders. Well, there's a certain particle size which impacts the powder flow. Then, of course, the shape. I mean, powder flow is always about friction in between particles. If you think of it, what happens during powder flow? Well, those particles, they're moving on top of each other, meaning there is friction in between the surfaces of the individual particles. And now you can easily imagine when sliding over particles with edgy shapes like that, there will be more friction as opposed to sliding over particles with a round shape. So this is how we have an impact here on powder flow. Same thing is for surface texture. It also accounts for the friction in the end. There might be some gas molecules adsorbed to the surface of the particles, also impacting powder flow. So those were now the given things of the powder where we, again, as a rheologist, can do nothing about it. But now when it comes to those imitation of the processes, which I mentioned, because it's always imitating a process, the external impacts are now the ones where we as powder rheologists can really play around to get representative results of my powder flow. For example, the applied stress. What amount of stress does my powder see when it flows? This heavily impacts the flow behavior of my powder. Then humidity. Just imagine now that these little droplets build liquid bridges in between the particles, in between the individual particles. They pull the particles together, meaning by that we increase the interparticle forces, which will in return have a huge impact again on the powder flow. So this is something we will have to consider during our powder rheological measurements. Then, of course, temperature. Just think that your powder is, for example, a polymer. You're transgressing the glass transition temperature of the polymer particle softens. Again, this will impact the friction in between the individual particles, which again changes my powder flow. Then also if my powder is fluidized or not, if I'm doing pneumatic conveying and so on. So those are now the external impacts where we as powder rheologists can simulate the process. In order to do so, we offer two different powder characterization cells. What I like to point out before I show them to you is now this categorization down here. We categorize them according to the load of the process we're trying to imitate. So for high load to medium load applications, we have a powder shear cell, classic for measuring flowability and so on, which we will see later. There we can play around with the humidity and temperature. And besides that, if we want to go into processes where we fluidize our powder, for example, fluidized bed reactor or pneumatic conveying, then we would use our powder flow cell, because here we can nicely imitate the fluidization process. We do that by simply, introducing pressurized air from down below into this powder flow cell. There we can also play around with temperature and humidity to a certain extent to really get representative results in terms of the powder flow behavior. This is what our two cells look like on the rheometer. Again, on the left-hand side, we have our shear cell in combination with a temperature device, and on the right-hand side, we have our powder flow cell where we can fluidize our powder by introducing pressurized air. So there are a lot of powder handling processes out there. That's why there's also a lot of methods for imitating those. Those are the well-established ones. We're not going into detail through them right now. I just want to point out that we cover all of these. We can look it up online. Classic things which are used a lot in pharma, for example, basic flowability energy, the BFE, we can measure that, as well as shear testing. But the focus today is definitely on the pharma industry. So let's see where exactly along the process route we can apply powder rheological testing methods from our rheometer. Well, the goal always is, right, you have some kind of active ingredient, and what you want to do is you want to dose it correctly into the system of a patient. However, most of the times this dose is really low. It's a couple of milligrams sometimes. So it doesn't go directly. We have to take a couple of different steps. We have to take a detour to get it there. So we mix it with an excipient, we blend it, get the mixture. Then if the flow is not well enough, we have to do granulation. Then still we have to dose it, maybe in a pouch like we're seeing here. The pouch is then opened again. We empty it, we dissolve it in water, and then it can be swallowed. This is one way of dosing it into the system of the patient. Another way would be that we start here again with our granulated powder. We tablet our powder, in this case into a fizzy tablet, which we can dissolve again, and again we can dose it to our patient. If we want to stay with tablets and not a fizzy one, well, we can also swallow this tablet directly. So we already see there are a couple of process steps until actually the active ingredient gets to my patient. So let's see where and which methods from powder rheology we can apply here during those process steps. So first of all, if I have a mixture of a powder, as we see up here, right, with different densities in this case, segregation is always something we have to watch out, right? There's not a lot you can do against segregation besides, minimizing your transport routes. However, what we can find out with our testing method is whether a powder has the tendency for segregation. Another thing here, of course, we can check is flowability. Maybe this mixture is already flowable enough. We don't even need to granulate it. If we have to do granulation, especially if we use dry granulation, well, compressibility is of interest for setting the process parameters for slugging, for example. If we then have our granulated powder, again, segregation testing comes into play, not because now of the different ingredients and different concentrations, because in this state, of course, the API should be evenly distributed in the granulated powder. However, what we often get from our customers is that if they are facing some problems with different particle sizes, because still you will not have the exact same particle size in your granulated powder, those particle sizes can segregate as well, which only then in a result will have or will cause issues when you're dosing it. So also segregation of a granulated powder is of interest when we're then dosing it, in this case into a pouch. However, if we're dosing it into a different container, we might want to check flowability and wall friction, because this will tell us how well the powder can be dosed, especially if I'm changing a supplier or if I'm changing a powder. Then if I'm really filling it into a pouch, the duration time test is of interest. We will talk about this one in detail later on, because this is not such a well-known. Basically, it tells me how long I will have to wait until I can seal my pouch. Once it is in the pouch, of course, I have to rip my pouch open and empty it again and put it into a solvent. What I want to have, if I'm pouring or I'm emptying my pouch here, I don't want any particles to be stuck on the pouch, on the inner side of the pouch. So with wall friction, I can look into the interaction between the powder and the pouch material. And of course, really important, if I'm dissolving something, this should always ring a bell if we're going to a distribution process in a solvent or dissolving, is permeability. Basically, there can measure how easily the solvent can enter into my powder cluster, so enter in between the individual particles. Great. If I decide to not go into a pouch and keep the powder as it is, I want to go into tableting. I mean, obviously this is compressibility. There I want to look into compaction behavior. I can take the process parameters then to transfer them to my tablet press to set the parameters correctly for my tablet punchers. Then again, I can measure here permeability. In this case, it is not because of the distribution, but actually when I'm filling the dye in my tablet press, I want to make sure that any entrapped air can also go out of the dye. I want to make sure my powder is permeable enough to not form voids inside my tablet. Then one last measurement method is, if I then go into fizzy tablets manufacturing, well, I want to look into the porosity of my tablet. It will tell me how easily it will dissolve. And this is something I can measure with our newest measurement method, which is envelope density. So, you already see there are a lot of measuring possibilities in this really, let's say, facilitated process route. So since time is limited, we will only focus on those 4, where I will give you concrete examples which we measured from customers. So we look into flowability, duration time, compressibility, and envelope density. Great. I will start with flowability because this is, well, the most typical one if we talk about rheology. Before we start in the concrete results, I would like to show you how we can measure flowability. Well, first of all, shear testing. It is typically used for medium to high load applications. Is for any dosing unit, this is the perfect testing method. Basically, what you measure is the flowability as a function of applied load. We will see that on the next slide. We can do temperature and humidity controlled measurements here. As opposed to that, also basic flowability energy, we can do that. This rather than goes into quality control measurements. Also here we can apply temperature and humidity. And the third version we offer for flowability test is the cohesion strength. What I like to point out here for cohesion strength is that it is really highly sensitive, and it has such a short testing time, which is smaller than 4 minutes, even smaller than 3 minutes if I think of it. Before we start now with the results, I would like to point out that our software is completely compliant with the 21 CFR Part 11, so it does all the audit trail and the access control. Okay, great. So in this case, I would like to talk about shear testing. Real quick, how does it work? Well, it measures the flowability as a function of the applied stress. How does it do it? Well, we calculate the so-called FFC coefficient, the flowability coefficient, And this always puts into relationship the applied stress, what stress my powder sees, and what is the internal resistance of my powder towards this stress. So what is the strength of the powder? So the flowability is always depending in this case on what stress the powder sees. If we now divide those two magnitudes, well, we get the FFC value, and basically, or most of the times, it is displayed as this. It categorizes the flow behavior between free flowing and non-flowing. The higher my FFC value is, as we see here, indicated by the FFC equals 10 line, the more free flowing it is. Because at this point, basically, the strength of the powder, or let's say the applied stress to the powder, is 10 times greater of what the internal resistance towards flow of my powder is, right? These flowability charts are really nice to look at, but actually the most important line here, which I like to point out, is this one, which is getting larger right now, because at this point, at an FFC value of 1, the strength of the powder equals the applied stress. So exactly here, we have an equilibrium of the strength and the applied stress, and there my powder will not flow anymore, just because it is too strong to be moved by the applied stress. This is really the most decisive one, because at this point, I can expect a lot of blockages or jamming taking place in my dosing unit. Great, so finally coming to the results, we tested neocitran, everybody knows it, right? You take it if you suffer from influenza, if you have the flu, you have some symptoms there. And what we measured there is the flowability at different relative humidity levels. We started with measurements at 7% relative humidity and went all the way up to really humid conditions, so to 95% relative humidity. And now looking at the results, we see at low relative humidity levels, we most likely will not have any problems with dosing because we are still in the easy flowing and free flowing area. However, once we increase the relative humidity further up to 65% and then even to those 95%, we can see that we are actually losing the ability to flow completely under the given normal stress. So this is something you will have to consider if you have manufacturing plants in different countries with different climates. Always keep in mind the surroundings of your manufacturing plant will a lot impact flowability, which in the worst case will actually cause your problem or your powder not to flow anymore. Great. This was it about flowability and dosing ability. Let's go to tableting, since this is such a big topic in pharma. We already mentioned that compressibility is the best choice here for imitating this process. What does the compressibility test do? Well, it's a really simple test. You press on your powder, you press, you increase your normal stress, and you see how the volume changes. This is what it is. You nicely see if you put more normal stress in terms of more weight onto a powder, the volume will change more. Typically, it is expressed as a function of the bulk density, meaning the bulk density increases the more you compress it, simply because, well, less voids are available in the powder and it is a denser material. Typically, we use this for packaging because I want to know how much mass can I fit into a defined container and a defined volume. However, when thinking of the tableting process, we nicely see that exactly this compression happens here in my tablet press. So it seems to be the perfect fit for imitating this tableting procedure. As I mentioned, what you get as a result is the bulk density as a function of the normal stress. If you're now interested in comparing, for example, the change bulk density at a given normal stress, if you want to compare this to the initial bulk density, when the powder is settled freely, then we can go on to Hausner ratio or Carr index, which simply eliminate the absolute value of the bulk density, and you will find out in percent how much this powder can be compressed. Great, let's go to the concrete results. So what we tested were 3 different microcrystalline celluloses, so one of the most used excipients in pharma. They were pure samples, however, they differed in their particle size distribution, and we can already see the differences here on the right-hand side. We tested up to 2.5 megapascal, and we see even though chemically seen those MCCs were completely the same, they show different bulk densities. What does this mean now? Well, my particle size distribution impacts definitely my compaction behavior, even though chemically completely the same. And if, for example, I am changing my supplier for what reason ever, well, most likely I will have to reset the parameters for my tablet press, and my punchers have to work differently. So please keep this in mind, that MCC1 must not equal MCC2, even though chemically seen the same product. Yes, I also plotted here the true density of MCC. Well, we see it's far above the one from the bulk density, which is of course expected because here we still have voids in between the individual particles. So that was really basic compaction behavior. What I like to point out is here that our rheometer is really such a high resolution tool that we can do in-depth analysis of compaction. So what we did here was that we solved for the Haeckel equation. I'm really not the expert for the Haeckel equation, but basically it tells you the 3 stages of compacting a tablet. And what you can do is that during the second stage of the deformation regime, you can put a linear graph in there, and if you solve for K and A, well, this will tell you how well a powder can be deformed, how its plasticity is. Again, please don't ask me about the Haeckel equation. I'm pointing out that our rheometer is actually able to have such a high resolution during the measurements to go really do also R&D measurements. Great. This already leads me to the next measurement method, which is the newest one in our powder rheology family. It is the envelope density. If you hear this term for the first time, please ignore this envelope at the end, at the beginning, because it is rather confusing. What it does really, it is a density measurement. We can measure the density of an object of a complex shape in our rheometer. Typically, density is always calculated mass by volume. Mass is easily determined via a balance. However, the volume of a complex shape needs to be measured. If you have a perfect cube, well, then you can calculate the volume, but most of the objects we are interested in have a complex shape, something like this. And there, of course, the volume has to be measured and cannot be calculated. Great. So how do we do that? Well, basically, we place it in our powder flow cell, and we compare the volume of a powder, which is first in there, to the volume of the powder after immersing this complex object in our powder. And the difference now between the first volume measurement of our powder cell and the second powder cell is now exactly whatever this new object pushed out, right? This difference here equals the volume of my object. If you think of it, you have a glass of water without any ice cubes, you add some ice cubes to it, then suddenly the level will go up, typical Archimedes principle, and this difference equals, well, the volume which was immersed into this displacement volume. So we're talking about porosity. How do I get from this density measurement to porosity? Well, I will have to combine it with gas pygnometry measurements, because this always gives me the true density of a material. So it takes away all the pores which are included in the envelope measurement, and then we can go to porosity. And this is exactly what we did with 2 different tablets. So we looked at 2 pills which actually had the same API in there, paracetamol, again against flu and cold symptoms. The one was a fizzy tablet, so you can dissolve it in water, and the other one was one that was swallowed directly. However, both had the same active ingredient in there. So let's see what we got out. Well, first the envelope density, where we had the mass divided by the volume, including the pores. We already had a difference here. Most likely this one is higher because it has a lot of sugar in here, which is heavy. Then we measured the true density, meaning we calculated the density without, or the volume without the pores. There are also differences. And now if we take these 2 values, so the true density and divide it by the envelope density, we get the porosity value. And we nicely see the differences here. So the fizzy tablet has a much higher porosity than the tablet which is swallowed directly. And this is exactly what we expected, because this higher porosity actually eases or facilitates the dissolution in the water. Right. This already leads me to the last measurement method I would like to talk about today, which helps us to set the correct parameters for filling if I put some powder into a pouch or in another container. So this is a little teaser. Finally, we are talking about fluidization. We nicely see how we can fluidize a powder here in our powder flow cell, where the pressurized air is coming from down below. And this is what we will be using for determining the so-called deaeration time. Why is this of importance? Well, if you think of it, if you're filling now a powder out of a chute into a certain bag, into a container, right at the moment where the powder is leaving the chute or the container, it is being fluidized because it is, well, pushed against the air. And what I want to know before I can seal a pouch, this is actually what we measured, this normal hydrol powder. What I want to know is that the powder is not dust anymore. Everybody knows that if you fill a powder into a bag, well, there's dusty air left. However, if those particles causing this dusty air are still here, well, I cannot seal my bag flawlessly because those particles prevent that. So what I want to make sure is, I want to know how long does it take until the entire dusty air settles. And this is measured by a deaeration time. Basically what I do is I blow a lot of air into my powder, I stop blowing air into it, I don't want to fluidize it anymore, and then I measure the pressure signal underneath the powder bed. And once this pressure signal reaches a constant plateau, as we have here, well, then we know that the powder is completely deaerated, it is completely settled, we don't have any dusty particles anymore, and now we can take this deaeration time exactly for setting this as a waiting time before we can seal our pouch. Okay, great. Thank you. This was it. Feel free to ask any questions.
Thank you very much, Helena. So as she said, you are now free to join us to the F&A on Q&A button to ask your questions to Ms. Weingrill. So we have a question already here. So can one of your techniques also be used to measure rheology of wet granulates to characterize the granulation process?
Well, that's actually a good question. I've read a lot about wet granulation, and I know there is this theory, if you continuously add a binder and keep on stirring it, then at the maximum torque, you should have the perfect concentration of binder for wet granulation. For sure, we can measure it. If this theory is correct, we have never tested it, simply because we haven't had time, but I'd be happy to test it. I've heard about the theory. We for sure have, let's say, the hardware specifications to do it. So I'd say let's try it out.
Thank you very much. So maybe another question. For your envelope density measurement, is your sample destroyed after the measurements? Are there any health risks by the displacement powder? And the person asked, since I have worked with mercury intrusion before.
Okay, thanks for the question. No, there are no health risks. We purposely chose a powder which is completely safe to use. It's actually a really free-flowing powder, of course, and the sample is also not destroyed. So you're completely safe here. It's actually glass beads and nothing to worry about.
Okay, thank you very much. So since you mentioned that you can do temperature controlled and humidity controlled measurements, what are your limits here?
Actually, we cover a pretty broad range of temperatures. We can go down to negative 160 degrees with our shear cell and the small flow cell, and all the way up to 980 degrees, if this is of interest. I guess for pharma samples, everything will be gone by that temperature. But yeah, we can go up almost to 1,000 degrees if necessary.
Okay, perfect. Thank you. So if you have any questions, we'll use the F&A button, so then I can forward it. And I think maybe one last question, if we do not get any other. What is lower limit particle size of your particle filter?
Okay, great. So for our flow cell, when we fluidize, we have this filter up there, and actually there the lower limit is 5 microns. There we're 100% dust proof. However, if we go further down to 1 micron, it's 95% dust proof. So still pretty safe, but until 5 microns, it's completely tight.
Okay, thank you very much. So then I think we do not have any questions left, but if you have any questions afterwards, please feel free to contact us and then we will forward it to Mrs. Weingrill. So then thank you, Mrs. Weingrill, for your interesting presentation.